Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I'm pleased to have the opportunity to share some insights with you. Today, we will be discussing the qualification of GC columns. Over the course of this presentation, we'll explore what GC columns are, why their qualification is important, and the key aspects involved in ensuring their proper functioning. I look forward to guiding you through this topic and hope you find it informative and engaging. Let's get started. Let's take a moment to explore what GC column qualification really means. Essentially, it's about demonstrating that a specific GC column consistently performs reliably for its intended analytical method. This isn't just a one-time check. It's a thorough demonstration to ensure the column's performance is up to standard. Now, why does this matter? Well, qualification helps us ensure the accuracy of our results, meaning the data we generate is trustworthy. It also supports precision, so our measurements are consistent over time. Additionally, it confirms the specificity of our method, ensuring we're accurately detecting what we intend to. Most importantly, proper qualification supports regulatory compliance and maintains the integrity of our data. In a regulated environment, having validated performance isn't just good practice, it's essential. So, as we move through this presentation, keep in mind that qualification isn't just a technical requirement. It's a vital step to ensure our analytical methods are reliable, accurate, and compliant. Let's take a closer look at the regulatory landscape that guides our analytical work. First, we have the ICH guidelines, which are globally recognized standards. For example, Q2, R1 focuses on the validation of analytical procedures, emphasizing the importance of ensuring our methods are reliable and reproducible. Then there's Q14, which deals with the development of analytical procedures, helping us optimize and understand our methods from the ground up. Moving on, USB standards also play a crucial role. USB 621 specifically covers chromatography, and it's applicable to gas chromatography when referenced. So, it's vital that we align our practices with these standards to maintain consistency and compliance. Additionally, regulatory agencies like the FDA and EMA have clear expectations. They emphasize qualification as a key part of the analytical method lifecycle, which includes ongoing system suitability testing. This means we need to continuously verify that our systems are performing correctly throughout the entire process. Overall, understanding and adhering to these guidelines and standards is essential for ensuring our analytical methods are robust, compliant, and fit for purpose. This regulatory framework ultimately supports the integrity and quality of our products. Let's take a closer look at the scope of GC column qualification. This process applies in several key situations. First, when we introduce a new column lot, it's essential to verify its performance before use. Second, after a column has been stored for a period or has been inactive, qualification helps ensure it still meets our standards. Third, if a column is transferred between different instruments, qualification confirms it functions correctly in the new environment. Fourth, anytime there are changes in the column type or its dimensions, qualification is necessary to confirm consistency. It's important to note that this scope is distinct from routine system suitability checks, which are ongoing and more general. Overall, the goal is to ensure that the columns we use consistently deliver reliable, high-quality results across these specific scenarios. Let's take a closer look at the key parameters for qualifying a GC column. First, the column identity is essential. This includes the column name, the type of stationary phase chemistry used, and the manufacturer. Knowing this helps ensure we're selecting the right column for our specific analysis. Next, we consider the physical characteristics. This involves looking at the length of the column, the internal diameter, or ID, and the film thickness of the stationary phase. These factors influence the separation efficiency and resolution. Temperature limits are also critical. Every column has a maximum temperature it can handle, so understanding these limits helps prevent damage and ensures optimal performance. Finally, inertness and deactivation properties are vital. They determine how resistant the stationary phase is to active compounds in our samples, which affects the overall stability and reproducibility of our analyses. All these parameters are interconnected and play a key role in qualifying a GC column, ensuring we get reliable, accurate results every time. Now, let's take a closer look at the column documentation and specifications. First, you'll notice the importance of clearly labeling each column and including the manufacturer certificate. This ensures traceability and compliance with quality standards. Next, the batch or lot number is essential for tracking production details and managing any potential issues that might arise later. We also recommend documenting the specified operating conditions, such as optimal temperatures and flow rates, to guarantee consistent performance. Safety and handling instructions are equally critical. They help prevent accidents and ensure proper use of the materials. Moving on, the qualification strategy overview provides a structured approach to validation. The typical workflow starts with protocol preparation and approval, which sets the foundation for the entire process. Then, visual and physical inspections are performed to verify the condition and integrity of the equipment or materials. Following that, system suitability and performance checks are conducted to confirm everything functions as expected before proceeding. We also evaluate the system using a standard test mix, which helps ensure accuracy and reliability in real-world conditions. Documentation and reporting are vital. They provide a comprehensive record of all activities and results. Finally, the qualification conclusion summarizes the findings and confirms whether the system is qualified or if further actions are needed. Overall, these details are crucial for maintaining quality and consistency throughout your process, aligning with our overarching goal of ensuring reliable, compliant operations. Now, let's talk about system suitability. This is a critical step to ensure our method is performing as expected before we proceed with analyzing real samples. First, we need to prepare a standard reference mixture that's appropriate for the specific method we're using. This mixture serves as our benchmark. Typically, it includes components like volatile organic standards, which are essential for calibration and detection. We also include critical pairs for resolution assessment, which help us verify that our system can clearly distinguish between closely related compounds. It's important that the injection conditions we use for this standard mixture mirror those of our actual method. This consistency ensures that the system suitability results are truly representative of the method's performance during routine analysis. By following these steps, we can confidently confirm that our system is ready, reliable, and capable of producing accurate, reproducible results. Let's now take a closer look at performance parameters. These are the key metrics we use to evaluate how well a system or process is functioning. They help us understand whether we're meeting our objectives and where improvements might be needed. First, we focus on parameters like efficiency, which tells us how effectively resources are being utilized. Then there's throughput, or the amount of work completed in a given time frame, an important indicator of productivity. Another critical aspect is reliability, which measures how consistently the system performs without failures. And don't forget about response time, which affects user experience and overall satisfaction. By monitoring these parameters closely, we can ensure our operations are optimized and aligned with our strategic goals. Remember, these metrics are not just numbers. They tell a story about performance and guide our decision-making process. So, understanding and tracking these parameters is essential for continuous improvement and maintaining a competitive edge in our industry. Let's talk about data evaluation. First, we compare our observed results against the acceptance criteria we've set. This helps us determine whether our data meets the expected standards. We use statistical measures like the percent RSD, which looks at the relative standard deviation for both retention time and peak area. This gives us a clear picture of the consistency and precision of our method. It's important to document any trends that fall outside of our specifications, as these out-of-specification trends could indicate issues with the column or the process. The ultimate goal here is to assess whether the column still meets its intended purpose, its fitness for purpose, so we can be confident in our analytical results. Now, moving on to test execution. We start by equilibrating the GC oven and column, ensuring everything is stable before running samples. First, we run a blank injection to check the baseline and make sure there's no contamination or drift. Next, we run our standard test mix to confirm the system's performance. Performing replicates, usually around 5, is crucial for reproducibility. During these runs, we record key metrics such as retention times, peak areas, resolution, and other relevant data. These steps ensure that our chromatography system is performing reliably and that our results are accurate and consistent. This process ties back to our overall goal of producing high-quality, trustworthy analytical data. Now, let's talk about troubleshooting common issues, specifically focusing on requalification and monitoring. Requalification is something we need to do under certain circumstances to ensure our system is still performing optimally. For example, after long periods of inactivity, it's wise to requalify because conditions might have changed, and we want to verify everything is still in good shape. Similarly, if there have been significant changes to our method, like adjustments or updates, we should requalify to confirm those changes haven't introduced any issues. Also, if we notice damage to the column or if its performance has altered unexpectedly, requalification becomes essential. Beyond these specific cases, it's good practice to set up periodic checks, say monthly, to keep an eye on the system's health. During these checks, we can trend key parameters such as plate counts, resolution, and retention times. Monitoring these parameters over time helps us catch subtle signs of deterioration early. This continuous monitoring and timely requalification are crucial parts of maintaining system reliability and ensuring consistent, high-quality results. They tie back to the overall theme of proactive maintenance and quality assurance in our analytical processes. When it comes to best practices, using the right installation tools is absolutely essential. Things like column cutters and wrenches are designed specifically for these tasks, and using them helps ensure a safe and precise installation. Next, it's important to condition the columns as required by the manufacturer. This step helps maintain their integrity and performance over time. Whenever applicable, always use guard columns. They provide an extra layer of safety and protection, which is critical in preventing accidents or damage. Ensuring gas purity and proper flow is another key point. This is vital for the system's efficiency and safety. Any compromise here can lead to issues down the line. Finally, documenting all changes and maintenance events is crucial. Keeping a detailed record helps with troubleshooting, future maintenance, and compliance. These best practices tie back to the overall goal of ensuring a reliable, safe, and efficient system. Let's take a closer look at the documentation section of our qualification protocol. Here, we outline the objective, scope, and responsibilities to ensure everyone understands their roles and what the validation aims to achieve. We also include the standard test mix details, which specify the parameters and tests performed during qualification. Next, we define the acceptance criteria. These are the benchmarks that determine whether the results are acceptable or not. The results summary provides a consolidated view of all the data collected, giving us a clear picture of performance. We also include raw data and chromatograms, which are essential for transparency and traceability. Calculations and comparisons help us analyze the data, verify consistency, and ensure compliance with standards. Moving on, the conclusion and status tell us whether the system is qualified or not qualified based on the data. If needed, we also include recommendations, such as how often requalification should be performed to maintain ongoing compliance and performance. All these elements together ensure thorough documentation, supporting a robust validation process that aligns with overall quality and regulatory expectations. Let's wrap up by emphasizing that column qualification is truly essential for ensuring reliable and reproducible GC analysis. When we properly qualify our columns, we can be confident in the consistency of our results, which is critical for making sound decisions. It also plays a key role in maintaining regulatory compliance, as authorities often require well-documented methods and validated equipment. Additionally, a thorough qualification process enhances the robustness of our method and improves data quality, helping us identify potential issues before they impact our results. It's important to remember that all of this must be documented carefully, with adequate justification and clear acceptance criteria, so that our processes are transparent and traceable. Finally, adopting a risk-based and systematic approach when qualifying columns ensures we're prioritizing efforts effectively, focusing on what truly matters for our specific application. Overall, integrating these principles helps us uphold high standards and achieve consistent, reliable analytical outcomes. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. You can contact us through the details provided, and we'll be happy to assist you. Thank you so much for your time and attention today. We truly appreciate your engagement and look forward to supporting you moving forward.